Okay, we have a question from Madeline Woods for Jeff. Do you think there would be a similar effect of pH on the settlement of other bivalves like mussels?
Yeah, that's a good question. So mussels typically settle on hard substrates rather than in sediments. So the settlement of a bunch of different benthic marine organisms has been investigated in the context of ocean acidification. And it seems very species specific. I can't speak off the bat relating to mussels, but there is some research that shows that ocean acidification can affect the byssal attachment strength of mussels. So mussels lay down adhesive byssus when they're settling and they're attaching to their substrates. So the compromised attachment strengths of these byssus can certainly affect, I would think, recruitment as these mussels are trying to make a go when they're really, really small.
And that is a great segue to promote the October webinar, which will be given by Emily Carrington, talking about the byssal strength of mussel, the strength of mussel byssal threads. So we have another question from Joseph Kunkel. Are there any reasons why lower calcification would result in behavioral changes?
So if you remember when I showed the ocean acidification slide, I showed 3 different processes that happen as CO2 dissolves into ocean. So the CO2 content increases, which we call hypercapnia. The hydrogen ion concentration increases, which we usually measure as a decrease in pH. That's what we call acidification. And the combination of these effects can lead to carbonate undersaturation or lower carbonate material for shell building. So what I actually think is happening with the burrowing behavior, when I showed the slide showing the way that this happens, or the mechanism that burrowing happens, we think it's a neurological one. So it's a physiological one. So yes, the shell is going to dissolve if they settle and burrow into low pH sediments. But it's not necessarily this dissolution of the shell that's causing the behavior. It's something different. And what we think it is that the increase in hydrogen ions interferes with the proper physiology of the animals. So the acid-base physiology of these animals. And they have to somehow physiologically compensate for this effect. And to do that, what ends up happening at this GABA receptor is that the flow of ions changes, and that is what causes the altered behavior. So the dissolution in the behavior, while the behavior can affect whether or not a clam dissolves, it's not necessarily that the dissolution is causing the behavior, it's something else.
Thanks, Jeff. We have a question from Monica. How does macroalgal cover, for example, ulva, affect the pH?
Yeah, so macroalgal cover we would predict would lower the pH. So as the macroalgae dies, that's going to cause a lot more organic degradation, which causes these microbes, again, to use up a lot of the oxygen and create this CO2. So we didn't specifically look at, I've never actually looked at what the pH is in macroalgal mats. I will say that our site that had the lowest pH anecdotally had the highest abundance of ulva on the surface. And there have been previous studies that have showed that these macroalgal mats can significantly decrease the oxygen in sediments as well. So yeah, I would say that increased algal mats would decrease the pH and cause undersaturation. But at the same time, again, I had mentioned the need to look at other abiotic factors like oxygen that are also changing at the same time.
Great. Another question from Bill Mook. Do you have any ideas on how your work relates to Brian's? It seems like he's saying that pH is not the issue in Freeport.
Yeah, so that's one of the reasons that I had mentioned at the end of my talk, the need to look at the effects of pH when predator abundances are high. So when you look at our sites, we really didn't capture any predators at those sites. If we had of, our results may have been different and may have actually corresponded with what Brian is saying. So I mean, we can't quantitatively compare how many predators were at each site. But I would say, yeah, when there's a lot of predators present, I would predict that, yeah, predation would probably override the sediment acidification effect. The other thing to consider in that is the time scale at which we were looking at these effects. You know, the sediment acidification effects that we were seeing were happening on very short time scales. Really, we're showing these happening on the time scale of weeks. So, you know, low predation, short time scales, maybe sediment acidification matters. And again, at the end of my presentation, I had mentioned the need to look at sediment acidification at longer temporal scales or longer time scales to really see where it has an effect and whether or not it actually matters.
Okay, and a follow-up to that from Sarah Randall. How did you determine that the field locations where you did your experiments had low predator abundance? And she points out that the research that the Down East Institute has done has shown that it is the small, tiny, and hard to see green crabs that are causing high levels, around 99% of mortality on the recruited clams.
Yeah, so at our sites, we had pitfall traps that we had set out prior to doing the experiments. And then we did capture some, but not many of the small green crabs at any of the sites, really. One of the big differences, so I'm not sure in the sites that Brian was looking at, the extent of the intertidal zone may play a role there. Up in the Bay of Fundy, the tides are crazy. At our sites, you know, we have a 4-hour window to sample, but we're talking about mud flats that are kilometers long. So the predators at high tide may not have to come in as far as we were sampling. So we were sampling fairly close to shore, 20 meters from the shoreline. Predators may simply not have to come in that far at our sites to get their food at high tide, just because of the extent of these mud flats and that the food could be available, you know, at the outer edges of the low tide mark.
So I have a question of my own to follow up on what you just said. Have you looked at all at the change in the pH of the surface sediments as the tide goes out and comes back in, so through a tidal cycle?
We have, and we see high variance, like super high variance. So we didn't really pick up a trend necessarily.
Okay.
Typically, the pH was a little bit higher. I mean, this is, I mean, a super sort of very, very slight trend. But we did see higher pHs as the tide was going out, and then pH decreasing as the tide was low. Whether or not that's a real effect, I'm not sure. But the sites that we were working at are really muddy for the most part, except for one. One had fairly coarse grains, and that's where we saw the highest pH. So I think that a higher overlying water pH, this is one of the hypotheses I have, is that when the grain size is high, there's more penetration of the overlying water, which has a higher pH, and that can help the pore water stay higher. Whereas at muddy sites, or really muddy sites, there's less penetration of that overlying water into the pore water, and the pH stays relatively low.
Okay. So we don't have more questions for Jeff at this time. Let's check back in. Oh, I see that Brian is no longer on the panelist list. Emily, have you been able to touch base with Brian?
Yep, I think it looks like it's an issue on his end. He can no longer hear us either.
Okay.
I asked if he wanted to log back out and log in again and see if that works. Otherwise, we can just continue with any other questions that come in.
Okay. So Bill has asked another question. I may have missed this, but do you know if Brian's shell treatments resulted in higher sediment pH? I'm wondering about the size of the shell particles. So I believe that Brian said there was not a significant difference in the pH based on the shell treatments. But he also said that the netted treatments had a lower pH. So, yeah. And I think that after Brian's audio cut out, I think he was getting into talking about the size of the shell particles just based on the images that he was showing. And another question for Jeff. Does the susceptibility of the GABA receptors to pH vary among bivalve species?
So far, we really don't know. We haven't really investigated that fully yet. So I can really only think of 3 studies that have looked at this GABA idea. Mine with the bivalves. There was some follow-up work in our lab by an honor student that we had look at this affecting Carophium, not bivalves, but these little amphipods that live in the mud. Up here in the Bay of Fundy, they get to extremely high densities, tens of thousands of individuals per meter squared up here. But they live in U-shaped burrows in the mud. And the work from one of my honor students there showed that their burrowing frequencies were negatively affected by sediment acidification too. That is, that they didn't burrow into low pH sediments, and that GABA was or at least there's evidence from the GABAzine experiments that mechanism was happening in Carophium as well. So they are the only 2 species with sediment acidification that I can think of that have looked at this GABA-A receptor. So if anybody wants to do other experiments with different species, email me. I'll be happy to show you how to do it. They're very, very easy and relatively cheap experiments to run. So if anybody wants to do them with more species and investigate this, I'd be more than happy to help out and give some pointers. And there is a third study with a snail species, not one that burrows, but one that jumps to avoid predators. It's a tropical snail. And what they found was with ocean acidification or water column acidification, we can call it, their jumping behavior was negatively affected by acidification. And again, with the GABAzine, it restored that normal jumping behavior under high CO2. So it seems like it's a pretty wide-ranging mechanism. We just need to do more tests with more species.
And we have a question. What species is the jumping snail?
I can't. Gibberlosis or something like that. I'll have to look it up. Yeah, it's a conch from Australia.
Sarah Randall has followed up on Bill's question about the sediment size, and she said that they deployed a variety of shell sizes and types in 2016. So up to the point where Brian's audio cut out, he was talking about experiments from 2014 and 2015. So we hadn't heard about this yet. And she's saying 1 millimeter clamshell, 5 to 10 millimeter clamshell, 15 to 20 millimeter clamshell, 1 millimeter oyster, and then also granite as a control for habitat, as granite would not buffer the sediment. And then additionally, controls to those experiments. And so Bill wonders, were the lower pHs in the netted treatments in the range that resulted in less recruitment or burrowing in Jeff's work? So I am going to ask Sarah if she recalls the lower pH levels in the netted treatments and could respond in the question field. And then we can compare that to the pH range that resulted in less recruitment from Jeff's work. So we'll wait a moment to see if we hear. Okay, so Sarah says that more clams survived under the nets, which were lower pH. We were wondering if you could remind us what the pH under the nets were, so that we could compare those to the ranges in Jeff's experiments where he observed lower burrowing rates. Okay. And Sam Sidelecki points out that Brian suggested that the pH was lower in the netted regions due to increased respiration from the trapped organisms. So this suggests that it may still be unclear if the crushed shell impacted the pH of the sediment pore waters. Thank you, that's a great point. And that was a question that Sam had been planning to ask to Brian. So unfortunately, since it's 2 p.m., which is the end of the webinar time, and since we have not had Brian join us again in this webinar, unfortunately, I think we are going to miss hearing the end of his presentation. This webinar will be posted on the NECAN website, and we will talk to Brian and ask if he would be willing potentially to record the ending of his webinar at a later date so we could post that to the website as well. The conversations of questions and discussion could be continued in the Ocean Acidification Information Exchange website, which would be a great opportunity to further these discussions. If you are not already a member of the Ocean Acidification Information Exchange, I recommend that you consider joining because it is highly frequented by academics, industry members, resource managers, and there are excellent discussions from interpretations of results to planning out experiments that could be very informative. And then we have one final question from Monica, who's asking if Brian's work is published. And I don't know the answer to that, but I will find out, and if so, we will send the links to you. So I apologize for these technical difficulties, but thank you so much for these great questions at the end, and we will certainly try to get the end of Brian's webinar up on the NECAN website. Thanks so much.